So yeah, thanks for having me here. I'll just begin today by acknowledging the Gadigal people of the Eora Nation, the traditional custodians of the land which I reside today, and pay my respects to their elders, past and present. I'm in the MEEP lab, and Professor Nate Lowe is my primary supervisor, so he'll know all about this research. This is his part of his research on cockroaches and termites. So we've been working on some cockroach genomes. And so we're focusing on this family up here, Bleberidae, but just to give some context, cockroaches are a really interesting and super diverse group, about 4,500 species. And termites are a type of cockroach, as Nate explained this morning, and there are about 3,000 termite species. And so over the past few years, 5 or so years, there's been a lot of really great genome research on cockroaches, with some really interesting findings. And displayed here are some of the genomes that have been sequenced and assembled. So your Blatella germanica, or German cockroach, and Periplaneta americana, American cockroach. So everyone's probably quite familiar with these two. We see them around the house and around the gutters and around the capital cities. And then there's quite a few, there's 3, maybe more termite genomes, a few coming out soon, I think. And so of particular interest of this genome work is the evolution of termites, because they're a really interesting group to sit within cockroaches. And in particular, the evolution of their eusociality. So that's why I'm in this symposium, because we've sort of tried to focus a bit on this. And it's quite a difficult question to tackle. So that's why researchers are now looking at genomes to sort of help answer this. But previous genetic studies or genome studies have looked at these two cockroaches as urban species. They're omnivorous, they do really well in the urban environment. And why do they do so well? Are there convergent adaptations within the two species? But as you can see, there's a lot of cockroach diversity where their genomes have not been sequenced. So we aim to address this, and we've sequenced and assembled 3 Bleberidae genomes. So these are natives, arid adapted species, and I'm a wildlife geneticist. I'm new to entomology. I've mainly worked on birds, but in my very biased opinion, these guys are stunners, and I've grown to, I get to hold them in Nate's lab and interact with them a bit, but yeah, compared to your American German cockroach, they're very, very likable cockroaches. And so why are we studying these 3 Bleberidae? Well, for a few reasons. Primarily what I've already said, it gives evolutionary context to the genome research that's already been performed on cockroaches and termites. So what does adding extra diversity within cockroaches change about our understanding of cockroach and termite evolution? And what extra inferences can we gain by looking at Bleberidae? And also there's a slightly different but connected part of the genome project is looking at some really interesting convergent adaptations within Bleberidae, which I'll discuss in a tick. So again, adding these 3 genomes provides some evolutionary context to look at especially termites and the evolution of their eusociality. So what genes are involved in that? So are there pheromone complexes, for example, and what genes are involved in caste determination within these complex social groups of termites? And we also wanted to look at some sex-specific genes. So cockroaches, like most male insects, don't just breed and then die. They often breed a few times. And termites, some termite species have taken this to an extreme. They can live up to 20 years. We also wanted to look at, again, revisit the evolution of American and German cockroach. So previous studies have looked into their chemoreception and detoxification and immunity, because they are such successful invasive species. They're omnivorous, they're very hardy. So we wanted to revisit that. So adding Bleberidae, how would that make a difference to these studies? Well, and a good example is if a previous study found that there's this special gene that they found within termites but not in an American and German cockroach, they might think this gene has something to do with termite evolution. It might be what makes termites termites. But if we found this same gene in all or any of the Bliberidae species, that sort of changes the whole context of that gene. So maybe it arrived in an ancestor of cockroaches and termites and was lost in German and American cockroach. So it sort of changes the whole story. So this is why adding the diversity is so important. And yeah, as I said, we wanted to also explore some convergent evolution phenomena in this group. So this is a figure from one of Perry's papers. I'm not sure if she's in this symposium, but basically the story is this soil burrowing cockroach within Bliberidae. This soil burrowing phenotype has evolved independently multiple times in Bliberidae from this wood feeding cockroach. And this has happened multiple times in the last 15 million years. And you can see the phenotypic differences between them. It's very obvious. So the soil burrowers have wing loss, reduced eyes, obviously very different behaviors. And it's a very complex phenotype to evolve. So it's amazing that it's involved multiple times. So we wanted to try and get more to the bottom of this using genomes. And then, as I've mentioned, there's a number of urban traits in the German and American cockroach that have independently evolved. Now, they haven't necessarily evolved to the urban environment. They've just evolved and enabled them to flourish in the urban environment. So we wanted to find out what genes were involved in these convergent adaptations, and if this is a case of parallel convergent evolution, where the same genetic machinery is used to create these phenotypes, or whether it was different genomic mechanisms, like your shark fin and your dolphin dorsal fin, that they've arisen through very different genetic mechanisms. And this sheds light on whether this convergent evolution is predictable or stochastic. And it just gives clues into evolution more broadly. So it's a really cool study system. So just to summarize the aims of this sort of genome research, we've got looking at the evolution of termite eusociality, looking to see if there's anything in Bliberidae that pops out as quite interesting. Looking at the convergent evolution of these soil burrowing compared to the wood feeding cockroaches, and then the convergent evolution of these sort of urban type traits. So to assemble a genome these days takes a lot of data, usually a lot of different sources of data. So these are STLFR, TELSEQ, they're all different types of genomic data. And I won't go into too much detail. But another thing that needs to be considered is the computer resources need to process these data. So for each genome assembly analysis, some of them are really computationally intensive, and each compute resource is better suited to certain analysis. So basically what we've had to do is decide what analysis we want to run, what genome assembly analysis we want to run, and then identify what compute resources were best to run that analysis. And it's quite tricky. Like, a lot of these compute resources listed here can't handle some of the analysis we run, but maybe one specific one can. So we had to run different steps of the assembly at these different supercomputer centers. And I mean, this is, again, I won't go into too much detail, but we had different types of data and different amounts of data for each species. So I used very different genome and assembly pipelines. And usually the more the data types, the better the genome. Yeah, I won't go into this, but I needed to show you because this is where all the work has gone into so far. And then these are sort of the results of these genome assemblies. So a bit of a boring slide, but these are important results for a genome study. So the first two metrics here measure the continuity of the genome. So the less fragmented, the better. So we want to assemble this genome into as big a chunks as possible, ideally chromosomes, which is typically not achieved. And then this N50 sort of is the median conti or scaffold that we've assembled. So it's 3.2 million base pairs compared to 11.4 thousand base pairs, for example. And then these next three metrics measure the completeness of the assembly, a measure of how complete the gene set is. And I guess the main takeaway from this is that the G. dilatatus is by far the best assembly we have. The other two genomes are much more fragmented, although the missing data, for example, for Panacea cribrata is not too bad. But we just, and they are still useful for some of these analysis, but we just need to take this quality into account when interpreting our results. And then, so with all this data, how do we find interesting genes? So what genes contribute to the evolution of eusociality in termites, for example? And it's with great difficulty. It's like looking for a needle in a haystack. So there's obviously tens of thousands of genes, and we might be trying to find one gene, or we might be finding a few connected genes doing something we're interested in. But luckily, these days, there's really powerful developments in bioinformatics that is making it much easier to find this needle in the haystack. It's incredible what bioinformaticians have developed in the last 5 to 10 years. So we can use these methods to look for interesting signatures of evolution and see what genes pop out. So one of these sort of types of analysis, and this is to investigate signatures of convergent evolution, is looking at parallel evolutionary rate changes. So an example here is on the right of a study looking for genes associated with hairlessness in mammals. So all the orange branches here are mammals with no hair or not much hair. And so we're looking at all these orange branches, are they doing the same thing as one another, which shows convergent evolution? So are they all longer than the average tree, or are they all shorter than the average tree? And it shows rate changes. So if all these orange branches, for example, are longer than the average tree, it suggests decreased evolutionary constraint due to loss of function at the gene, or it's suggesting directional selection to alter functionality. In contrast, if they're all shorter than we expect, or shorter than the average tree, might mean they're evolutionary important, so they've been conserved. So this just sort of, the genes that pop out from this type of analysis are sort of, like the needles we're trying to find in a haystack, where the genes are hay, I guess. And we've done this sort of analysis for all our common orthologs across all the cockroaches we've been looking at, plus some other insects, plus some candidate genes we're interested in, like genes that have been associated with wing loss, for example. We wanted to see if they popped out in species that have independently lost their wings, to see if there was these convergent signatures. And another thing we looked at is identifying groups of genes that have expanded or contracted. So the important distinction here is orthologs versus paralogs. So orthologs are homologs that derive from a speciation event. So this frog gene with the mammal gene, and then paralogs are homologs that derive from a duplication event. So this star here, the ancestor of dogs and humans, there was a duplication event within the genome, and then subsequently those two genes formed orthologs as they diverged within the dog and the human. So basically, we're looking at the trajectory of these different duplication events among the lineages and seeing where there's been a gene duplication event, how many duplications there have been, or has there been a gene contraction event where genes are lost. So we only have quite preliminary results at the moment, but some interesting things. So this is an example of this, the convergent evolution rate changes that I was discussing earlier. And this is focusing on the N. hanni and G. dilatatus convergent evolution of soil burrowing. So these sort of were some hits within many hits that sort of had parallel accelerated evolution. And for example, the top one, which is displayed in this figure, which shows the rate changes, the top gene was involved in regulating osmosis and stress. So these are arid adapted insects. We thought this was maybe worth exploring further. And then, so this is looking at it from looking at American and German convergent evolution, and some of the genes we found had got to do with digestion. So they're omnivorous species. So this top one, which is displayed in this figure as well, so the microsomal triglyceride transfer protein, that's associated with lipid digestion. So it's being conserved because this rate is slower than the average tree. The average tree being 0 here on the figure. So yeah, maybe this lipid gene has something to do with their omnivorous diets. Again, these are just genes that are worth exploring further. Doesn't mean that this gene's definitely associated with convergent evolution in these species, but it's sort of candidates for exploring further. And I've only got a minute left, but I'll quickly wrap up the gene expansion contraction results. So this figure doesn't tell us too much other than at each of these nodes and taxa, how many genes have expanded in green, how many gene groups have expanded in green, and how many gene groups have contracted in red. And looking at this more closely, in the termite ancestor, there's some interesting genes that expanded, including ubiquin-related genes. And ubiquitization is a form of post-translational modification, so it might help with caste differentiation. We found some sex chromosomal type genes that expanded, so it might have something to do with caste differentiation or weirdly long-lived male termites, and some insecticide stuff that might be interesting. And then expansion related to urban environments, there was lots of digestion type genes, so maybe that's got something to do with the omnivory. And your cytochrome P450s, we found a few expansions there, which might be related to detoxification in an urban environment, and some receptor families that were found in previous studies. All right, that's all from me. Thank you to everyone in the slide. Lots of knowledge and really great people helping out with this, collaborators, and obviously Nate. And thank you for all the funding and resources that you need to do this sort of genome research.
All right, thanks, Kyle. That was really nicely delivered and very clear. I think we'll leave questions until the discussion